Hi everyone, today we are going to learn about growth. Why growth is important in living organisms? Multicellular organisms add more and more cells to form more tissues and organs as they grow. The growth and development of living organisms are not the same things. Growth is the increase in size and mass of that organism, while development involves the transformation of the organism as it goes through the growth process. There are three subtopics in growth: 10.1 Measurement of growth, 10.2 Types of growth curve, and 10.3 Growth pattern. But for subtopic 10.3, we will be discussed in the tutorial class. So we start with the first subtopic 10.1 Measurement of growth. So in this subtopic, you should be able to define growth and explain how growth is measured. What is growth? So growth is a permanent and irreversible increase in size of an organism over a specific time through cell division, cell enlargement, and cell differentiation. What is the cell division? Cell division is the process by which a parent cell divides into two or more daughter. Cell enlargement, there is an increase in the volume of cytoplasm and the cell organelle. Without cell enlargement, no organism can continue to grow. While cell differentiation is the cells which are less specialized become more specialized by certain modification and changes in the cell. The process of growth is begin from the zygote stage to adult stage, and the process may continue throughout life or cease at maturity. Growth can be measured by using a certain parameter over a period of time, and the parameter chosen must be appropriate to the organism whose growth is to be measured. For an example, height is appropriate for human but not trees, which is a growth of root cannot be measured. Measurements of growth in unicellular organisms, especially microorganisms, is done by measuring its population growth. Example, Paramecium grow or reproduce colonially via a process called binary fission. It is different in multicellular organisms. The growth is monitored by measuring the changes in quantitative parameters. The growth parameters include biomass, length, width, height, surface area, volume, and cell number, and it is measured over a period of time. Example, mass of organ, length of an insect, height of plant, and surface area of leaf. There are two types of measurement of growth. We can use a size as a measurement of growth, and another one is biomass. So we look first to the size. Okay, for the size, we can use the length, height or width, surface area, and also the volume. But there are advantage and disadvantage by using the size as a measurement of growth. The advantage of by using the size as a measurement of growth is it is easy and quick, no need to kill the organisms, and can be measured continuously to observe the growth. But the disadvantage by using the size as a measurement of growth, it is a only measures in linear dimension. What's the meaning of linear dimension? Linear dimension is a measure in one direction. and does not consider growth in other direction or dimension. Example, we use height as parameter to measure growth of human over a period of time, and it happened in one direction. Other than size, we can use the biomass as a measurement of growth. And for biomass, it can be divided into two types. The first one is a wet or fresh mass, and the second one is dry mass. Wet or fresh mass is a mass of an organism under normal condition that is without removal of body water content. And for dry mass, is a mass of an organism after removing all its water content from its body by drying. We look to the wet or fresh mass first. The advantage of using the wet mass to measure the growth is easy to measure because it requires little preparation of the sample. And it also does not cause injury to an organism. And the same individuals can be used to monitor growth of an organism over a period of time. But disadvantage by using the wet or fresh mass is it does not measure true growth because it may give inconsistent readings. Why? Because water content can increase or decrease regularly depending on the organism's water intake. The advantage of using dry mass as measurement of growth is accurate measurement of the amount of organic matter present. Why dry mass of an organism is the best indicator of growth? Let me explain to you. This is because dry mass weight of an organism exclude the fluctuating water concentrations in a biological organisms or material which is usually present in the fresh mass of the organism. But dry mass still have the disadvantages, which is the organism has to be destroyed or killed during drying. The growth of the same specimen cannot be measured continuously, cannot be used to monitor growth of an organism over a period of time, need a large number of genetically identical specimens grown under similar conditions to measure growth to obtain a representative reflection of growth, and also the time consuming.